Hello my dear students, welcome back to a new video of Sunanda's tutorial. Today I will discuss about the breakdown of triglycerides. It is the part 2 video of lipid metabolism. In my part 1 video of lipid metabolism, I have already discussed about how the triglyceride synthesis occur in the living cells. So let us start about the breakdown of triglycerides. At first, the breakdown of triglycerides or triglycerol is required to obtain energy from fat molecule. They act as respiratory substrate to release energy in the form of ATP. When levels of carbohydrate are low in living cell, generally carbohydrate act as the respiratory substrate. It break down to give energy or supply energy, but the case where when the level of carbohydrate is low, then the fat or triacylglycerol act as respiratory substrate to provide energy in the form of ATP to the living cells. In plants, it mostly occurs during germination of seeds to provide energy through beta oxidation. By the process of beta oxidation, the oxidation of fatty acid occurs and soluble sucrose to young seedling through glyoxylate cycle. Then the triacylglycerol is first degraded by hydrolysis. Hydrolysis means the breakdown of the bond by the presence of water molecule. So triglycerides first break down by into two components. One is fatty acid, the second one is glycerol. The released glycerol enter to glycolytic pathway or glycolysis as dihydroxyacetone phosphate or DHAP. The other constituent fatty acid are oxidized by beta oxidation to form acetyl coenzyme A. Acetyl coenzyme A, which is used by TCA cycle to generate ATP. The acetyl coenzyme A actually enter after the link reaction during the respiration process to the Krebs cycle. So after the formation of acetyl coenzyme A, it will be entered into the Krebs cycle and ultimately generate ATP. Hence, breakdown of triglycerides can be discussed under two phases. One is phase 1, hydrolysis of triacylglycerol to glycerol and fatty acid, then oxidation of fatty acid to ultimately release the energy. Then hydrolysis of triacylglycerol or triglycerides, which is known as lipolysis, and fate of glycerol. At first, the breakdown of glyceride is initiated by the enzymatic hydrolysis. In plants, it occurs in spirosome. It is a special types of oleosome or oil body which store fat and oil and are rich in hydrolytic lipase enzyme. In animal, the lipase activity is commonly seen in cytosol of adipose tissue, pancreas and intestine. The fat or triglycerides are hydrolyzed in presence of water and enzyme lipase to diglycerides, then to monoglyceride and finally to glycerol and fatty acid. So at first from triglyceride, diglycerides are formed, then monoglyceride form and finally glycerol and fatty acids are released. So the first reaction, the glycerol is first phosphorylated by the addition of ATP to form glycerol 3-phosphate in the presence of the enzyme glycerol kinase. Then in reaction 2, glycerol 3-phosphate oxidized by NAD plus to yield dihydroxyacetone phosphate in presence of glycerol 3-phosphate dehydrogenase. So it is a two-step process ultimately producing the dihydroxyacetone phosphate. Then oxidation of fatty acid. And in this two-step, previously discussed two-step, explain the how the glycerol breakdown occur or what is the fate of glycerol. Then oxidation of fatty acid, which is the second component of triglyceride. Fatty acid released from hydrolysis of triacylglycerol is broken down by stepwise oxidation of acetyl coenzyme A, which in turn usually undergoes complete combustion of carbon dioxide and water via Krebs cycle and electron transport chain to generate energy. So at first acetyl coenzyme A will be formed. Then from acetyl coenzyme A enter into the Krebs cycle, forming the carbon dioxide and water molecule, and then to electron transport chain to generate the ATP or energy. During the process, removal of carbon atoms begins either from alpha or beta or omega carbon position, basing on which it is named as alpha oxidation, beta oxidation or omega oxidation. Beta oxidation is the most common and major pathway of degradation of fatty acid. So it is either alpha oxidation, beta oxidation or omega oxidation. So at first removal of carbon atom begins. If it removes from this position, then it is known as alpha oxidation, and this is the beta position. When the carbon removed from this position, this is known as beta oxidation, and when the terminal carbon is removed, then it is known as the omega oxidation. And the nearest carbon atom to the carboxyl group is denoted as alpha, then the second one is beta, and the last carbon atom is designated as omega. And according to the removal and their position, the name of the cycle is decided, whether it is alpha oxidation or beta oxidation or omega oxidation. In my next video, I will upload or discuss about the details or detailed pathway of alpha and beta oxidation. So it is all about the breakdown of triglyceride. It is the part 2 video of lipid metabolism. Tomorrow I will upload the part 3, in which I will discuss the alpha and beta oxidation pathway. So stay tuned and enjoy my videos like this, and please like, share and subscribe. Thank you.